Thank you so much, Leonel. Thank you also for allowing me to participate in this course. That's very nice of you. It's been a great course. I've been learning a lot. That's really nice. And exactly, I work at, as Leonel was saying, in the Institut Pasteur, but in Paris. And my background is in physics. I actually studied 2 semesters in Buenos Aires, close to here. So I'm also really happy to be close to there. And now I work in a so-called image analysis hub. That's how our group is called. And basically, we are a core facility that is dedicated to performing image analysis services. And that covers kind of a relatively large range between very low threshold services that we give to people in just some short consultations, and goes over kind of maintaining infrastructure so that people can do image analysis in an autonomous way. And we also perform kind of the development of small pipelines for people, so special developments or also longer ones, basically. And this goes all the way up to developing software tools that people can use for their image analysis, either autonomously or also tools that help us help people better. And these would basically exceed then kind of collaborations that we have people in helping them on their individual projects. But yeah, exactly. There would be tools also that we hope will be of more general use. We're a relatively small group. We've been coming from the optical microscopy platform. And so we kind of split out from there. So there's a bit of a problem that we're losing some of the contact to the optical microscopy facility, but at the same time, this allows us to focus more on actually on some of the analysis that is also that was born from a strong need at the institute for people to need help with analyzing their data. So I wanted to quickly go through some of the tool development projects that we have in the group. So actually, one of our main, in terms of what we do in the group, often boils down to very similar use cases where basically people look at an object. Most of the times they're cells, but they can also be kind of subcellular structures or anything else. And they want to basically segment them, identify them, detect them somehow. And then every once in a while they want to track them over time. And what often people do is to then extract some features from these objects. Of course, this can be either shape features or something regarding the statistics of the fluorescence that is detected under these objects. So one of the tools that was developed for that in the group is a TrackMate that you might have heard of. Exactly, and this has really leveled up in the last years because of the advent of all of the deep learning methods for segmenting cells. So for tracking, you really need to have a very good segmentation of your objects at first. And that is really made possible by methods such as StarDist and CellPose that you might have heard of. And these are also incorporated into these plugins. So as a facility, we don't develop AI algorithms ourselves so much. We try to more make them available to people. And this is one of the efforts on kind of making them available within the Fiji plugin. Another project is the Java Deep Learning library, which basically tries to make. Sorry, I'm a bit, as you can see from the barbijo, the mask. And exactly, and this library basically makes AI models that are pre-trained on kind of these days that are very generalizable, that work on many types of data that are pre-trained. People upload them to a so-called bioimage model zoo. And then using this library, you can actually apply them on your data within Fiji or IC, which is another Java-based image analysis software. Oh, that's very nice. I'll put it here, and thanks a lot. Yeah, exactly. So another aspect of our group basically goes about making analysis more computationally efficient. That is in terms of running things on the GPU, but also in terms of dealing with large data. So that kind of we have a lot of developmental biology projects where people look at entire embryos and they look at large tissues over long periods of time. And people then very quickly get into problems by having too many object representations in in their tracking and segmentation. And basically then there's also developments to make these things more compatible with large data. And along these lines, some of the things that we often, so I want to speak to you today about one of the tool developments that relates to this aspect when we get kind of very large, sorry for that, very large data sets that often come from multi-positioning and microscopy, when basically very often kind of you acquire your data in tiles, because either the camera sensors are not large enough to cover the full specimen, or for other reasons. And then you would also have the case of multi-view acquisitions, where you don't only go acquire your data in different positions, but you also kind of go around your sample. That has some advantages that we'll discuss in the following slide. Exactly, but all of these things have in common that you first need to register your data. So you basically have all of the tiles and these so the tiles over positions and over different angles. And you know more or less how they relate to each other over the stage and over how the microscope is kind of the information that it gives you. But this is never perfect, and you always end up kind of with an only approximate type of overlap, and then you need to still, with an automatic method, try to overlap these different images. So this you will always have to do, and then you want to maybe fuse these different types of images. This is not always needed. I think Bruno was already saying that there's sometimes you don't actually want to fuse your data, because you might actually be fine without doing so. But sometimes you do want to do that for segmenting different types of structures at different scales, and also for in the case of light sheet imaging that I want to quickly speak about. That is a very common use case for this that I think Lionel was speaking about yesterday. So about if you remember light sheet microscopy, where you basically, which is very good for, as I was saying, imaging kind of large specimens, and that is very common not only in cell biology but also in developmental biology. And what people can have, what can happen to you is that if you put your sample here in the middle of so you put it in the microscope, and if you look at this top projection of your data, what will happen to you is that this is the part that is in front of the camera, and this is the part that is away from the camera. Because of scattering in the medium and absorption and many processes, you basically lose a lot of your image quality as you go down in the sample. And what you can do then is, and this is true for light sheet microscopy, but this could also be, and what you can then do, this slide is a bit strange, you can rotate your sample within the light sheet microscope, or within even a confocal microscope. You could rotate it, and in this way, if you acquire your images at different angles, you would have access to more data. And so then you get access to kind of the regions that before were completely dark, where you weren't collecting any photons from. But what is also good about this is that in this view we have a different kind of a different visualization of this. What is also true is that from different views you get different shapes of your point spread function. And so, for example, if this is the zebrafish example again, you get an elongated point spread function this direction here, and an elongated point spread function this other direction. And if you fuse, and then there are ways basically to fuse your data to using multi-view deconvolution, where you basically take into account all of these different PSFs for the different directions, to then basically estimate what could be the underlying distribution of your fluorophores. And if you just average, you basically get a blurred version of your images. But if you perform these type of multi-view deconvolutions, you can improve on all of these complementary views that you acquire. And then from this example, you get something like this on the right. Yes. So, and while there is software for this in Fiji, there's something called a Big Stitcher that has been often used, and that is being used a lot. But however, kind of there's lacking maintenance for this, and people need to basically have different type of registration and fusion algorithms available when things start to fail. And we're developing a Python software for this, which lets you do exactly this kind of image registration, fusion, and in general stitching steps. with diverse algorithm implementations, because in Python, all of these things are already there. They just need to be also again stitched together. And this is then kind of a stitching software in 2D and 3D. The first thing that we do there is to, especially when the data is very large, to convert the images into a different file format. So these are kind of considerations of more on the software side in a way, but you need to convert your files in order to, if you look at kind of input TIFF files, for example, then these would show you kind of the data all in one large chunk of the data. But if you want to visualize your large images, you would basically want to have file formats that allow you access to individual blocks of the data so that you can more efficiently visualize these. So this would be a first step that we would perform. And then we would make use of this representation of the data that is very localized. So where you basically have access, very efficient access to each of the parts. And we would do the step of image registration. So first, kind of when you remember all of these different imaging modalities, and we perform a kind of a group-wise registration. First, we determine what things need to be registered against which others. So this is kind of, we create a view adjacency graph. This is called, and then we determine which different tiles to register against each other. And then we go into the different pairs and try to only use the overlap region to actually perform this registration. And then the same is for the image fusion. This is a bit of a bit maybe too technical slide, in the sense that kind of once we know all the transformation parameters, we actually can then transform all of the images and fuse them together. For example, using multi-view deconvolution, or also a logic in the case of Bruno, for example, where you would say, I would always want to keep the first image, for example, that has been acquired, and for example, throw away data that was acquired after that. In the case of wanting to, for example, discard bleaching when you want to stay relatively quantitative. So this would be a function that we plug into here, for example. So and this allows us to reconstruct very large data sets. For example, many users use the Zeiss, so many people use a common multi-view light sheet microscope is the Zeiss Z1 or Z7 microscopes, and we can then basically take a lot of large 3 input stacks and reconstruct these in total specimens. Or exactly all types of different kind of 2D, 3D data sets. We're also kind of applying this a lot to cryo-EM maps. And exactly. There's an Apari plugin that we also developed for these things, where you basically load in all of your image layers, and then you add them to the plugin. And this is a very optimized approach, let's say, where you basically just have a register and fuse button, and you will see that kind of before you can see that here the overlap between the images, between the tiles is changing because we're optimizing it from kind of in this registration. Here you will see that the stage positions before were inaccurate, and we're kind of correcting them. And this is important very often for kind of further analysis things. And this is just another, I'm trying to be short, I'm almost done. This is just another video that is basically showing the same, a bit more interactive in an Apari, where you load in a lot of TIFF files into your viewer, and you just at the beginning arrange them in a way that you're saying this could be more or less the way that I want to stitch my data. And then the plugin basically does the rest, in the sense that it takes this as an initial configuration of all the tiles. And then you can see that it's basically, this is it. So this is like a 3D data set. Okay, the video is loading a bit slowly, and here you can kind of fuse all of these things together. And you can see that kind of the transformations have been corrected for it before. Okay, and with that, that was just a quick kind of overview bit of the different plugins and things that we do in the Image Analysis Hub. And I would like to thank the people there, and thank you a lot.